Thank you very much. Good afternoon, everybody. Our first presenting company this afternoon is Histogenics. And if you caught my panel this morning, you'll have already met Adam Gridley, who's the president and CEO. Now, Histogenics is a regenerative medicine company that's developing NeoCart to repair defective cartilage of the knee. However, the company sees this really as a first product of a regenerative medicine platform. So Adam is going to present a couple slides, and then we'll moderate the Q&A. And if there are any questions in the audience, don't hesitate to raise your hand. We'd love to keep this interactive. And Adam, I'll turn it over to you. Thanks for being here.
Excellent. Thank you. And thank you to ARM for having us. We're pleased to be here. This is, I think, the second or almost third year that we've been here. We think this is a great event. And certainly, there's common goal here in the industry to advance regenerative medicine. And as you mentioned, this is a platform opportunity, but it's one that's been under development for many years. So I'll give you a little bit of background on that. Before we get started, we are a public company, so we have a number of forward-looking statements that I would note you go to our SEC filings to look at the risks and uncertainties. Getting right to what Histogenics is, we are focused on fixing knee cartilage defects. This is the holy grail of orthopedics. Physicians, patients, and insurers are all looking for better alternatives. And we think this is a disruptive late-stage cellular therapy, really a symbiotic combination of cells, biomaterials, and engineering that creates fully functioning cartilage tissue. And this is different from any other products that are in development that we know of, and certainly in late-stage clinical development that we're currently going through. From what we have seen, this is a very simple surgery. It's a rapid recovery, and we've seen excellent safety because naturally this is an autologous therapy with the patient's own cells. Over the years, a number of companies have tried to develop products. I think the biggest challenge is this is an avascular area, so if you have a cartilage injury, the body has no natural propensity to heal this defect. And so unlike bone and tissue, there's no sort of inflammatory response. And this is one of the single biggest challenges and causes of late-stage osteoarthritis. Now, the market is, many of those is in the room. It's healthy, active adults, and so I think that's something that also differentiates from the much more well-known osteoarthritis market, where you're dealing with a lot of pain, you're dealing with limited function. These are the weekend warriors that end up with an injury and have significant loss of pain and function and really don't have a lot of other therapies. So what we're trying to do is bring those patients who are sitting on the sidelines and come in and actually have a therapy that works quickly, and works over time. We are running what we think is the world's leading phase 3 study right now under the FDA guidance documentation. We talked a little bit about it this morning. The regulatory environment has evolved, but it's taken a long time. The first product that was launched came in about 15 years ago, or actually 20 years ago at this point, and it took the FDA 15 years to put out guidance documentation. We are running a trial that is almost 90% enrolled, following those guidance documentation, looking at pain and function. We expect the trial to be enrolled in about 2 months by June of 2017. We reported that we are 214 of the 245 patients required back in March. And then we are the only company also with a 1-year endpoint. That's because we're the only ones that are making tissue ex vivo, and we believe are the only ones that are going to be able to show that difference at 1 year. What we'll see in the next slide is a little bit more on the data and the benefits, but this we think is going to be a huge barrier to entry, one of the world's leading trials, and we're very pleased to be able to report the outcomes of that next summer. That potentially takes us to an approval in 2019. It's a huge market. As I noted, there's about 500,000 procedures currently. About half of the patients are sitting on the sidelines doing nothing because there just aren't good therapies, and we're looking to change that. And because of some of the challenges in manufacturing over the last couple of years, we've been investing not only in the raw materials that lead to NeoCart, but we're making NeoCarts every day. This is a biologic, it's complex manufacturing, it's very expensive, but our goal is to be able to make sure that we don't have any surprises at the BLA review from a compliance perspective, and we're thinking about scale every day. How do we bring the cost down so that we can sell this affordably to our surgeons, and so they can use this in a large body of patients? What's nice is that allows us then to develop additional cartilage products for other indications. We're doing some work not only in autologous therapies for today's knees, but you could potentially look at hips, you could look at shoulders, you could look at elbow. And then we're doing some additional work on allogeneic stem cell sources so that we may be able to think about one-step procedures in the future. We've got a great management team in place. A company came out of the Brigham and Women's Harvard Medical School. We've got a leading scientific advisory board, great long-term investors, and just couldn't be more pleased with the outcomes of the work that we've done thus far. But it's been a long pathway. So in summary, the benefits that NeoCart provides through this ex vivo manufacturing process are dramatically accelerated recovery. We just reported some results from our phase 1 and phase 2 studies where we saw highly statistically significant improvement in pain and function as early as 3 months. And that's because we're making a piece of tissue. And for those that can see this, this is a NeoCart. We make this outside of the body. We don't have to rely on the body to make new tissue, which we won't do normally. We do it ex vivo. And so when the patient has this put in via a proprietary glue, best thing for them to do is get around and walk. And so we see great return to function and pain much earlier than any other products or the competitive procedure that is out there. Surgeons love it. It's a 20, 30-minute procedure offered by virtue of this glue. And then the clinical results are just outstanding. We had highly statistically significant data in a dual threshold responder analysis in our phase 2. Our phase 3 is just a larger version of that. And so with only 30 patients, we are seeing highly statistically significant data. So we think it's a pretty de-risked program. And of course, most importantly, it's got a great safety profile. Final thing that I direct you to is a couple of photos. These are always impressive. You can actually see that as early as 3 months in the one photo, this is a patient who was doing yoga at 3 months in our phase 2, tore her meniscus unrelated to NeoCart. So we're like, great, we get a picture. You can see it's fully integrated. What's more impressive is the surgeons often will say, well, wait a minute, my yoga patients are never doing, or I'm never back doing yoga at 3 months with microfracture. They do that with NeoCart, and we think that's going to grow the market. Patients don't want to wait 12 months for a product that doesn't work very well and then have it fail again in 2 years. So we think it's a great opportunity.
Good. Well, that was a very tight and very thorough overview of the company. And I want to ask a couple questions because let's start really with the market here for NeoCart, autologous cartilage repair, or microfracture. Tell us a little bit about microfracture. It's been a little long enough from lunch that I think we can safely discuss
we can talk about it a little bit.
But how many of these are there? What is a patient's expectation of response from microfracture, kind of gold standard today?
Sure, that's a great question. And microfracture is a surgical procedure where the surgeon will go in and drill into the subchondral bone. And what they're doing is creating holes where they're trying to recruit mesenchymal stem cells that will hopefully magically differentiate into hyaline articular cartilage. Problem is, most of the time, you just create scar tissue. It's equivalent to sort of cutting your hand and then sticking it under a faucet for 12 months and hoping you're going to get a scar. It's really hard to do that. What's critical for microfracture, though, is a very extensive rehab, and it's the Steadman-Hawkins protocol that was developed many years ago. So to get the best results, often in healthy, younger patients, low BMI, they've got a very prescriptive rehab program, 4 to 6 hours of continuous passive motion machine per day to be able to get the best results with microfracture. The problem is that you've got extensive rehab. Patients aren't very good about it. It takes about 12 months to get back to where you were, if you ever get back to where you were. Let's say you're a runner or a marathoner. And then the literature has now seen over about 20 years that at about 2 to 3 years, got a 30% failure rate. And that's because you're just making scar tissue. So that's the standard of care. And that's why it's so hard to enroll these trials, because most patients will do that and say, well, wait a minute. You got a 30-year-old person who's chasing her kids around. She can't afford to do that rehab. And if it's just going to fail and she's going to get OA anyway, why bother?
Although some would argue chasing your kids around may be continuous.
That actually may help. Who knows?
So our next presenting company, Marisol, has canceled. So you now can actually badmouth them or say anything you want about them. They're not going to be here.
No, not at all.
But I want to next kind of transition because Marisol did, and it was actually back when Genzyme pioneered this approach of autologous chondrocyte implant with Carticel. Very difficult procedure, trying to suture on periosteum over and then inject, essentially liquid expanded chondrocytes. So how is NeoCart different than MACI, which is their newest product, which is injected onto a collagen form and glued in, What's the primary difference? I think you're going to explain it's this ex
Sure.
vivo.
Sure. So starting, that's when you look at clinical performance and the surgeon and the patient experience, it all starts that we're making ex vivo tissue. So that's where the biggest difference is. We're the only one with biomarkers of cartilage at the end of our 8-week process. We can show histologically DNA SGAG. We have fully functioning tissue ready to be withstanding weight bearing and ready to be glued in. So you have a very different piece of tissue versus MACI, which is cells on a scaffold. Effectively, they solve the delivery challenges of Carticel. It's a great product, and sort of as a starting point, we have great respect and hope they're very successful. So we're all in this together. It's a huge industry, and I think we can both participate. But that first starting point is so different, and we've got tissue that doesn't have to wait 8-12 months to make cartilage, it's already doing so. So then when it goes to the patient, the surgeon loves it because it's very easy to cut. All they have to do is trace and effectively glue it into that defect. Our proprietary glue is a combination of polyethylene glycol and collagen, which is effectively a super glue, and it only requires about 10 seconds to cure. So you also don't have to wait like you do for the fibrin glue that they're using, and it's a 20-minute procedure. So this can go to every surgeon that is doing microfracture, not just the KOLs who are doing Carticel.
Now, talk to me. So how many, is it 200,000, 600,000 microfractures a year? I think maybe 200,000 in the
Sure,
US?
that's about right. And the way that we think about it, we start at the total market of those that have potential cartilage defects as seen by insurance records via arthroscopic evaluations. There's about 1.2 million scopes that identify these defects. Only about 600,000 patients will actually do anything. So there's half the patients sitting on the sideline. Most of them are just getting debridement, which is I go in, I see I have a problem, I'm going to clean up the area, and that's pain control only, and they stop there. Then there's about 200,000 that will go through with microfracture. And then the remaining portion are grafts. After you've failed your first microfracture, you go for your second microfracture, you give up, and then you go to Carticel, and then you have a total knee. Yep. So, I mean, that's what we're trying to interrupt is you turn back time with this product and avoid all of those additional surgeries and avoid OA.
Great, that's helpful. Now, Adam, you talked about this dual function and pain endpoint in the phase 2. Maybe you can bore down a little bit more into that detail. What were the improvements that you saw? How's function measured? I know pain is typically measured by Koos pain scale.
That's correct.
So maybe you can walk us through those 2 endpoints and the improvements that
Happy
you saw.
to do so. So great memory. Yes, there are a number of patient-reported outcomes that the FDA called out in their guidance documentation. Koos pain is one of them. IKDC function is another one. And these are patient-reported outcomes, both subjective and objective outcomes that are validated and listed as endpoints that the FDA requests that companies use when they develop these trials. And years ago, it used to be MRI was the way that you would evaluate a lot of these orthopedic procedures. One of our doctors calls it MR-lie because it doesn't tell you anything about pain. And so FDA guidance said use both of those endpoints. We took it a step further, and we're doing a dual threshold responder analysis that you commonly see in cardiology, oncology, where you have to hit on both pain and function, clinically meaningful improvements, and then we count the percentage of NeoCart patients that hit on both versus the microfracture patients versus the mean of the scores across the entire trial. And that's very different because you can imagine that patients will respond to pain. So they will report low pain scores because they've restricted their function. So I don't get down on my knees, chase my kids around because my knees hurt. So the dual threshold responder analysis we think is the real leveler, and it really defines the qualitative improvement on both of those, not just one individually. We have to see about a 20 percentage point delta in our clinical trial for the phase 3, and we saw close to 50 percentage points in our phase 2. Yeah. Highly statistically significant, and keep in mind this is at 1 year. We're the only company that is going against microfracture at 1 year covered by our SPA. Yep. And that's because most companies have to wait for microfracture to level out and start to fail before they can show a difference at 2 years. Yep. We see it as early as 1 year. We think that's going to be a huge competitive advantage.
Yep, that makes a lot of sense. Now, maybe you can tell us a little bit about that phase 3 trial design. I know you're about to finish enrollment.
Sure.
We've got a 1-year follow-up, as you just said. So just maybe tell us a little bit more about that study, and obviously we'll be getting data next year. So.
Sure. It's a 245-patient, 2-to-1 randomization directly against microfracture. And so agency has been very clear that you have to compare against the gold standard, which we're doing and everyone else will do. Inclusion and exclusion criteria are actually intended to favor microfracture a bit. We know that microfracture does not work well in heavier patients or older patients, and so inclusion criteria is 18 to 59, BMI of less than 35. The biggest challenge for everyone doing these trials is you can't have a concomitant procedure. So when you blow out your knee, skiing, running, playing hockey, or just falling down the stairs, 60% of the time you have an ACL or meniscal tear as well. Now, in practice, you'd fix both, but for the clinical trial, you can't fix both because the FDA wants to know that the pain and functional improvement is because you fixed the defect. The trial started about 7 years ago. We had to stop to upgrade our manufacturing. We ran out of money along the way. Every company has had such difficulties enrolling these because of the narrow criteria, even though the market is very big. And we really kicked this back into gear about 2014, and now we're at 214 patients.
Great, that's awesome. And just for people joining us, our next presenting company, Marisol, has canceled from the conference today, so we're going to take a little bit more time to talk to Histogenics before starting with our next presenting company, Bone Therapeutics. So Adam, we talked a little bit about this at the panel this morning, but I kind of want to revisit this because are there opportunities for the 21st Centuries Cure Act to, I mean, obviously you're already well underway with phase 3, but how can that help you nonetheless?
So I don't know if it'll help us specifically because you're right, Ted, we are so far along, and if we were doing this prospectively in another era, maybe it would help us. In this particular case, what I think it will do is it symbolizes the partnership that the agency is taking with companies. We've seen it. I think Marisol has seen it. There's much more of an approach of letting good science drive decisions, having thoughtful discussions about what is reasonable for some of these biologics, particularly on the manufacturing side as well. It's clear that you have to have safety. There's no question whatsoever that even with the new act, safety standards are not going to change. But I think what you're seeing is sort of a sea change in the way that the agency is approaching this. And they are trying to bring what are reasonably effective therapies that are curative. Because keep in mind, we're turning back time, we're not replacing. We're actually trying to take patients back and prevent other issues such as OA. And if they're seeing such impressive clinical data, I think that we're seeing that they're more apt to work with folks. And I think that's good. Good. But we're not compromising anything.
Now, you talked about manufacturing and process improvements. Sure. Maybe give us a little bit of more background on how you can accelerate or lower costs. I mean, some of that will just happen with scale, obviously, but what are some other active things that you can do to drive down costs?
Sure. So a couple things, all of which we're doing, and we've really learned from some of the challenges in the industry. I sort of joke, we're not necessarily that smart. It's actually better to be a second or a third follower because we've learned from some of the challenges. So first is raw materials. We're using a collagen source, a scaffold, some very expensive biomaterials that probably would normally make up 40–50% of your cost of goods sold. We've brought all of those in-house, first for compliance reasons, for FDA reasons, but then also for scaling. So that's the first thing that we'll do to be able to bring costs down, and that's going to help enormously as we're doing several thousand or tens, 20s of thousands of units a year in the future. That would have really hurt. The other thing is going from autologous to potentially allogeneic sources, because then you're going to continuous manufacturing. Right now with autologous therapies, every product is a lot. Every patient is a lot. And we make NeoCarts every day right out in Waltham. And we're effectively in commercial manufacturing right now. Now that's challenging as a small startup, being a public company, spending a lot of money pre-commercialization. But it helps us be able to plan to scale up and scale out. And when you move to allogeneic in the future, you're then doing continuous manufacturing. You don't have all of the testing for every lot. You have the same testing for what may be 1,000 units versus 1 unit. So that's where there's a logarithmic opportunity to bring down costs over time. All stuff we're working on.
So you touched on allogeneic, and I'll probably finish with this unless there's any questions from the audience. But what is What are the challenges? The benefits, think, seem very, very clear in terms of moving from auto to allo. What are the challenges in accomplishing that? Is it finding cell type? Is it activity? What do you need to solve?
I think probably the biggest challenge for us in this particular indication, which is fairly unique, is really going to be figuring out the best regulatory pathway. In our particular case, we're looking at induced pluripotent stem cells as our allogeneic source, and that's really new. In Japan, for example, it's well sort of accepted, and there are in-human trials. But here in the US, the regulatory pathway isn't that clear. So that's where that partnership that I talked about, I think, will be critical. In our particular area, though, cartilage is a unique and probably one of the best places to go for allogeneic. It's immunoprivileged. There's already ample evidence in the literature of being able to use other donor tissues. And for the reasons that it's so hard to make cartilage, because there's no repair propensity, you've got a closed system, also make it a very safe area to go after. So I think the big challenge is regulatory. We already have proof of concept in less than a year. Yeah. But how do we partner with the regulatory agencies to show that it's safe? Because that's going to be most critical.
And that's maybe where the 21st Century Cure Act can come in.
That's exactly where it will help
really
for future applications.
Great. Well, I'm going to stop us there. We had the opportunity to go a little extra longer, and I think that was time well spent. So, Adam, thank you. I really enjoyed spending time with you today.
Likewise. Appreciate it.